Hello everyone, welcome back. We're going to be covering activity 7 today, which is looking at the kingdom fungi. Okay, so it's going to be very similar to how we discussed Proteus. I'm just going to give you guys some background information and you guys will draw the images, the microscope slide images in your lab manual, and you'll also answer some of the questions so that you can differentiate between the different phylas we will be discussing. So we already discussed the difference between the the kingdoms. We had the prokaryotic, which were bacteria and archaea, and then we discussed the eukaryotic, which are protists, plants, fungi, and animals. So today we're going to discuss fungi. So these are eukaryotic organisms. Fungi are not plants. They do not produce food through photosynthesis, and they lack chlorophyll. It might be you might think that they look a lot like plants, as shown in these two images above, but they are not plants. They actually are heterotrophs, and more specifically, they are heterotrophs because they secrete various digestive enzymes to decompose decay matter, as shown here, and they absorb this material. They are composed of a cell wall. So like bacteria and plants, they also have a cell wall. However, their cell wall is made of the polysaccharide called chitin. Um, as we discussed, the cell wall of many bacterial organisms, or the cell wall of bacterial organisms is composed of peptoglycan, and the cell wall of plants is composed of cellulose. So their cell wall is made of chitin. Chitin is the same, it's a very tough material. It's the same material found on the that comprises the exoskeleton of insects. It's also the exoskeleton of crabs. They are filamentous, and we'll discuss this later, and they do not have true tissues. So this is just kind of a recap. We said that fungi are chemoheterotrophs. They absorb via digestive enzymes. They can be decomposers, parasites, and symbionts. And then as far as cellularity is concerned, they can be both uni and multicellular organisms. So an example of a unicellular fungi would be yeast. In our lab manual, we mostly are going to discuss multicellular fungi. So this would be, examples would be mold and mushrooms. So in this image up here, you see the mushroom is made up of mycelium, which is all this network down here, and this is assumed image of mycelium. So mycelium is a network of tissue and is made up of smaller threads called hyphae, which is shown here. And these hyphae are zoomed in further, you would see the cell wall. And like we said, the cell wall in fungi is composed of the polysaccharide chitin. So the hyphae can can be two forms of hyphae. You can have the septate or non-septate. So here the septate hyphae has these cross-bodied walls. It's shown here, here, and here. And they are going to separate these cells. And an example of this would be penicillium. And then an example of non-septate hyphae. So the cell walls are fused. As you can see, there are no divisions here. So the cell walls are fused with many nuclei. The nuclei are shown in brown, and they have a common cytoplasm. And we will talk about an example of this, which is from the Phyllium Zygomycota, Rhizopus. And then these Fungi can form reproductive fruiting bodies, and this is what we think of as the mushrooms. So let me just go back here. So the reproductive fruiting body would be these mushrooms. So now going on to fungi reproduction, we're not going to cover this extensively in the lab. All you need to know is that fungi can reproduce both sexually and asexually. The majority of the cells during the fungal life cycle are haploid. They can reproduce sexually by fusing nuclei, and this is shown here. The nuclei will fuse in the plasmogamy. During plasmogamy, and this is all written out for you down here. So although there are many variations in fungal sexual reproduction, all include the following 3 stages. So during plasmogamy, the marriage or union of cytoplasm, 2 haploid cells will fuse, leading to the dikaryotic stage where 2 haploid nuclei coexist in a single cell. During karyogamy, the nuclear marriage, the haploid nuclei fuse to form a diploid zygote nucleus. And finally, meiosis takes place in the gametangia, organs in which gametes of different mating types are generated. At this stage, spores are disseminated into the environment. So it's shown here how they will fuse together and go through this cycle, but like I said, you don't really need to know that for the lab purposes. I do present it in all the slides just so you have a general understanding, but you do want to know that they can reproduce both asexually and sexually. Here showing those stages again. When they reproduce sexually, this is going to introduce a lot of genetic variation into the population of fungi. When they reproduce asexually, they are essentially just releasing spores. They could also be budding or going through fragmentation, but typically it's releasing spores, and these spores can be carried through for example, the wind or other animals and be spread out. This will release thousands of clones, and it's going to rapidly increase the spread of their progeny. So remember, these are not plants. They are not reproducing through seeds or pollen. They are reproducing through spores. So as you go through this activity, we're going to introduce 4 phylums, and these are phylas. These are classified on the basis of the spore. So we're going to look at the Ascomycota, which have ascospores, Basidiomycota, which have basidiospores, Zygomycota, which have zygospores, and Chytridiomycota, which have zoospores. So you really want to know these 4 different phylas or phylums and the spore type that corresponds to each one. So as you go through the lab manual, you're going to 1, sketch the specimens in your lab manual, and 2, answer the following questions. Where are they found? What is the name of their reproductive structure? It is listed here for you. How do they get their food? And list some common examples. So let's go through the first one. Chytridiomycota phylum. So these are found throughout wet soil and lakes. They can be decomposers, parasites, or mutualists. An example of a mutualist would be a chytrid that lives symbiotically in the digestive system of cattle or herbivores. An example of a parasitic or parasite would be, this is a disease, it leads to chytridiomycosis, and this affects the amphibian population, so frogs. It is an amphibian chytrid fungus. That disease is caused by the chytrid fungi Batrachochytridium. And here's just some information on this. This has been affecting the amphibian population since I believe that it really came to be known about in the 1990s, and you can see that these are a species of amphibians that are critically endangered due to this fungus. And then this is the surface of the epithelium of frogs, and you can see that it shows these discharged tubes of spores from the fungus. um, Obviously, when you handle these frogs, you're going to want to use gloves to reduce the spread of this fungus to other animals and, just protect the environment. So these are the earliest phylum of fungi to evolve. They were originally classified as protists. However, they are not protists, they are fungi. Their reproductive structures are called zoospores. Remember, there's an image here that shows this reproductive structure. And zoospores are motile and they use a flagella. This is an image of the phylum Chytridiomycota. Chytridiomycota. I don't know why I always have trouble with that word. So then you would move on to Zygomycota. So when you think Zygomycota, you want to think bread mole. Is a common example of Zygomycota. And like I said, you'll want to draw the image shown in your lab manual. There's images here that you can choose from as well, or down here. So you can draw these images in your lab manual. And here is a chart discussing how sexual reproduction occurs in this species, as well as asexual reproduction, or in this phylum, not species, I'm sorry. They are decomposers, parasites, and mutualists. Their reproductive structures are called zygospores. Zygospores form when two haploid hyphae fuse into a specialized structure called a zygosporangium, and both sexual and asexual life cycle. They have both sexual and asexual life cycle stages. And I just want to show you here that an aerial hypha produces a sporangium, and this is an example of asexual reproduction. An example of sexual reproduction is when this the mycelium grows and then the hyphas form, these gametes form at a tip of a hypha, as shown here. This leads to the zygospore that forms in between these two. So this is asexual, this would be sexual. And you see those in your lab manual. This shows it budding off the atrial hypha, and this shows it in between. You would go on to Ascomycota. I'll let you guys go ahead and do this on your own, but I did give you guys some information for each one, and I gave you multiple images just so you could really make sense of these. When you see Ascomycota, think ascus, meaning sac fungi. These are very diverse group. You have truffles, you have baker's yeast, antibiotics. We already discussed penicillin last week, which was discovered by Alexander Fleming. And then you also have these other very diverse types of Ascomycota. You have this one that would feed on wall paint, and one that is able to decompose jet fuel. These are mainly terrestrial, also in molds. And these are decomposers, pathogens, and mutualists. So here you see the ascus shown here and the ascospores inside. And that is also shown here for you. Here's some more information on them. And in your lab manual, you would draw penicillium. It talks about the ascospores within a sac for sexual, and then asexual would be the spores conidia, and it's shown up here. And then we talked how they could also reproduce through budding, so you have budding yeast shown here. Okay, so you will also learn about the phylum Basidiomycota. So this is referred to as the club fungi. And I'm just briefly going through this, but I want you to really sit down and look at all the information that's provided to you. But they're classified as club fungi because during sexual reproduction, spore-bearing structures called basidia grow along the tissues called gills. So underneath the fungi, you will see these gills. Here's a zoomed up image of the gills. These gills have these basidias, and these basidiospores form off of these basidias or basidiums. So you see these spores and they have this club-like appearance. So here you have your basidia and then your basidiospores forming off of that basidium and your basidiospores forming off of that. And once again here, I try to give you as many examples as possible so you can really make sense of it. These are mainly terrestrial. They are mainly decomposers, and common examples would be toadstools, mushrooms, stinkhorns, and shell fungi. So here I have an image for you to kind of look at the sexual reproduction of these. But remember, you can always use, sorry, I'm going to go back, these images to explain their sexual reproduction. So basidiospores for the Basidiomycota, which is what we're discussing right now. And then finally, you're going to watch some videos regarding fungal interactions. So these fungi, they live, they work in a, have symbiotic relationships. There are two examples given in your lab manual. One is with lichens. So lichens is composed of both fungi and cyanobacteria or algae. And then you are given another example, which is found between fungi and plant roots. So lichen is a mutualistic symbiotic relationship between algae or cyanobacteria and fungi. And you can see these algae are found in here, and the fungi is encompassing this algae to form lichen, which is shown here. So lichen is not one organism, but two that live together. And this is really important to give rise to life on land. And like I said, I'll post two videos that I would like you guys to watch regarding these mutualistic relationships. So mycorrhizae is another one that exists between the, these are not roots, by the way. So fungi do not have roots. I forgot to point that out. This is the mycelium. Remember, it is densely packed. It is not roots. But here you see the symbiotic relationship where fungi can give different water and mineral nutrients to plants or trees in this example. And they provide carbohydrates through photosynthesis. So photosynthesis byproducts would be provided to the fungi. There are two forms. There is endo and exto. This one is far more common, and in the endo example, the hyphae will not penetrate the root cells. And then in the exto example, the hyphae the hyphae penetrate the root cells here, and in the exto example, the hyphae do not penetrate the root cells. And that is the end of this. If you have any questions, please let me know.